everybody. This is Nafrit Sekho, and I welcome you on this platform, The Layers of Life Sciences, where we discuss all the aspects of life sciences in quite depth and detail, layer by layer. The objective is not just to help you in your post-graduation, graduation, and competitive exams, but also just creating the general awareness about the life processes happening around us. In the last session, we have started with the discussion of biochemistry. In biochemistry, we need to discuss all the biomolecules, their structures, and their metabolism. The first type of biomolecules which we have chosen for the discussion are the carbohydrates. In the last session, we also discussed that the building blocks of the carbohydrates are the monosaccharides. Then, thereafter, we discussed that what is the definition, which will be the chemical as well as the structural definition of those monosaccharides. The definition was that all the monosaccharides are either polyhydroxyaldehydes or the polyhydroxyketones. If the monosaccharides are polyhydroxyaldehydes, then they are referred as aldoses, while if they are polyhydroxyketones, then they are called ketoses. Today's session, we are going to start from the detailed discussion about the structure of various aldoses. Thereafter, we will discuss the structure of ketoses as well. So let's start this discussion with the structure of aldoses. Now, we know that aldoses are polyhydroxyaldehydes, which means that if there is a carbon chain, then one of the two terminal carbons will choose to form a bond with oxygen, which will be a double bond, right? So it is just the choice that which carbon chooses to. Now, rest of the carbons in this carbon chain, right, apart from this carbon, which is involved in the formation of carbonyl group, rest of the carbons in this carbon chain will fulfill at least one of their valencies with the help of hydroxyl group. So now the presence of carbonyl group at the terminal carbon makes this molecule an aldehyde, while the presence of so many hydroxyl groups on all the other carbons makes it polyhydroxy. So it is basically a hydroxyaldehyde, right? So this is basically polyhydroxyaldehyde. Rest all the valencies over here are fulfilled by the hydrogens, right? So as we have discussed in the last session, that carbons are tetravalent, so there must be the presence of 4 bonds around the carbon anyhow. Now, let's compare this general structure of polyhydroxyaldehyde with all the aldoses over here. Over here, you must also be able to notice that in all the molecules of these monosaccharides, there is a terminal carbon which is involved in the formation of the carbonyl group. So it means that is true that they are aldehydes. Now, notice all other carbons in the chain. You would notice that yes, each and every carbon in the chain is associated with one hydroxyl group, due to which every molecule has so many hydroxyl groups. So all of them are polyhydroxyaldehydes. That's why they are all aldoses. Now, we need to learn the structure of these aldoses. There are two reasons, but first one is more pertinent. Now, what are those two reasons? Number one is that when we will do the metabolism of carbohydrates, be it anabolism or catabolism, we will require to know the structures of these monosaccharides. We should be knowing that what was the original structure and how that structures undergo conversions, how it is converted to another molecule and which molecule they are being converted into. So in metabolism, we will, to understand the metabolism well, we should have a good knowledge about the structures, right? So that's why, first of all, be it aldoses or ketosis, if you want to have a strong hold on the metabolism of carbohydrates, we need to have good idea about the structure of various aldoses and ketoses. Second reason is that there are so many analytical questions which are asked from this part, which actually they all relate to just the basic structures of the monosaccharides. So if you would be having any, even a smallest idea about the structures of these aldoses, you will be able to answer them quite confidently. So these are the two reasons for which we need to learn. Although it may seem to be a huge task, and it may seem to be quite hard, but actually it is not. Like learning so many structures is not hard because there are so many general things, like there are so many common things present in all these molecules. So let me give you some tips and tricks that how to learn and or just have a good acquaintance with all the structures of these aldoses, and then we will go to the ketoses as well. So first step in just having the good hold on the structures of these aldoses is that you learn that which aldose falls in which category of aldoses, right? So for example, glyceraldehyde is basically a aldotriose. Why it is aldotriose? It is aldotriose because its carbon skeleton is composed of three carbons, while erythrose and triose are aldotetrose. Why they are aldotetrose? Because over here there are four carbons which are present, right? In each of them there are four carbons. So they are erythrose and triose are aldotetrose, while ribinose, arabinose, xylose, and lysose, all these four of them are aldopentoses because there are four carbons present in them. And allose, altrose, glucose, mannose, glose, idose, and galactose and talose, they are all aldohexoses because, as you can see over here, each of them are composed of six carbons. So once you know the names and in which category they fall, certainly you will get to know that this structure involves the carbon chain of how many carbons, right? Once you know that, at least you will be able to draw the skeleton of that molecule. For example, somebody asked me to draw the structure of arabinose. Now, I know that arabinose is aldopentose. It is an aldopentose, means pentose will shows that it is composed of five carbons. So what I will do, I will draw um, a chain of carbons which will be five in number, right? One, two, three, four, five. So this thing I just got to know just by knowing that in which category of aldoses this arabinose falls. So first thing is done. Now it is aldopentose, which means aldopentose means it is an aldose, right? Which means it is polyhydroxyaldehyde. So if it is aldehyde, then one of the two terminal carbons should be involved in the formation of the carbonyl group. Now, it can be this carbon over here, just a minute, this carbon over here or this carbon over here. It can be any of these two, right? The uppermost or the lowermost, because they are the terminal carbons, right? So carbonyl group can be present on this one or this one. But by convention, we always draw the carbonyl group on the uppermost carbon. And this carbon is always numbered as one. And the others are then numbered as 3, 4, 5, right? 2, 3, 4, 5. Now, this carbon number 1, if you will count, it's one of its valencies is fulfilled by binding to this carbon, then by this two of them are fulfilled by this oxygen, double bond oxygen. Now fourth will be satisfied by just one hydrogen. So the valencies of this carbon over here are all satisfied. Now we will move to the other terminal carbon, which is carbon number 5, and which is right now at the lowermost position in this vertical chain. So over here, as we have done in the general structure of polyhydroxyaldehydes, we know that apart from the carbon involved in the formation of the carbonyl group, rest all the carbons in the chain fulfill one of their valencies with hydroxyl group. So all these carbons, be it carbon number 2, 3, 4, or 5, all of them will be fulfilling at least one of their valencies with the help of hydroxyl group. So same thing will be done by this terminal carbon over here, which is carbon number 5. So now around this carbon, this hydroxyl may be oriented in this direction. If not in this direction, it may be oriented in this direction. Okay, if not in this direction, it may be oriented on the right hand side of the chain. It can be anywhere. It can be anywhere. It's choice, right? So for the time being, let's keep the hydroxyl group on the right hand side of the chain, but it's not important that it should be on the, for arabinose, it should be on the right hand side. No, it can be at any position, right? Now, there are rest two valencies, which will certainly be fulfilled by hydrogen over here, right? Now, the valencies of both the terminal carbons are well satisfied. Now we move to the interstitial carbons. Now, these are the carbons between the two terminal carbons, right? Now, we know that for all these interstitial carbons, there are at least two bonds around them. So another two bonds need to be formed with two groups. Now, one of, as it is polyhydroxyaldehyde, so one of that group needs to be hydroxyl group, right? And the second group will be hydrogen group. Now, I can do this thing that I may put hydroxyl groups, right? All the hydroxyl groups like this. And on the other side, we may put hydrogens. But I cannot do this. I cannot do this because it really matters that hydroxyl group is oriented in which direction around all these interstitial carbons. It didn't matter around the terminal carbon. On the terminal carbon, it may orient itself in any direction. But around the interstitial carbons, the orientation, or we can say configuration of hydroxyl groups is really, really specific for a particular molecule. Because if this orientation varies, the molecule changes, its physical properties, its chemical properties change. That's why, so now it means that all these interstitial carbons have a specific orientation for hydroxyl groups. Now, how to know that what is that orientation? For that, we need to learn these codes over here. You can see there are codes written at the top, right? Anyhow, let's just go to arabinose once again. The code for arabinose is 34, which is 34. Now, what this code is denoting over here, the code 34, what this code is denoting over here, it is denoting that on the carbon number 3 and 4, hydroxyl group needs to be on the right-hand side of the carbon skeleton, right? So what about carbon 2, carbon number 2? If it is not mentioned in the code, it actually means that it over here, the hydroxyl group is on the left-hand side of this chain. On the alternative positions, we can place our hydrogens, right? So this is basically the structure of arabinose. This is basically the structure of arabinose. If instead of arabinose, I would have chosen ribose to be drawn, again, I know that ribose is again aldopentose, which means the carbon number will remain same. Then it is aldopentose only. It is not ketopentose. It means it is again the carbonyl group will be on the terminal carbon only. So the basic structure for both of them will remain same. Now the change will just be with respect to the orientation of the hydroxyl group. Now, for that, as I've told you, we have the codes. For ribose, the code is 234. 234. So, and that means that even on the second carbon, the the hydroxyl group will be oriented on the right hand side, and that's why the hydrogen group will go to the left hand side. Same thing is when we are doing allose. Allose has there are 6 carbons, right? So interstitial will be just 4. So if hydroxyl group is on right hand side on all the 4 carbons, then its code will be 2345. So, and for then altrose comes for that, just on the second carbon, the position is altered, then it codes remains 345. Then in glucose, the position um, around 3 is also altered. So it is 245, 245. So this is how we can learn the codes, and learning these codes will actually help you to draw all these structures, to know about these structures in quite detail, and you would be much more confident when you will be dealing with all such questions. So this is about aldoses. Now we need to go to ketoses. Now, ketoses are what? Ketoses are basically polyhydroxy ketones, right? The chemical definition of ketoses is that they are polyhydroxy ketones. So polyhydroxy ketones means, polyhydroxy means there must be so many hydroxyl groups present, right? Polyhydroxy ketones. Now, which actually means that if there is a carbon chain of 4 carbons, it can be 5 also, it doesn't matter, right? So now this is a ketone, right? Ketoses are ketones, which means that the carbon involved in the formation of carbonyl group cannot be terminal. It needs to be interstitial. It can be any interstitial carbon basically, but it is generally one which is attached to the terminal carbon, right? So it can be this one or this one, right? So this is the ketonic group now, because the carbonyl group is present on the interstitial carbon. Now, rest all the carbons in the polyhydroxy ketones or ketoses satisfy at least one of the valency with the help of hydroxyl groups. So, due to which there are so many hydroxyl groups present, that's why this molecule is called polyhydroxy. And because interstitial carbon is bearing the carbonyl group, that's why it is called ketone. So these are polyhydroxy ketones. Just like as polyhydroxy aldehydes, rest all the valencies will be satisfied with the hydrogens. No hydrogen on this carbon. It's all the valencies are already satisfied. And there will be 2 hydrogens over here. So this is basically the polyhydroxy ketone. Let's compare this general structure with structures drawn over here. So again, interstitial carbon, carbonyl group, interstitial carbon, carbonyl group, interstitial carbon, carbonyl group on interstitial carbon, and you can see over here also they all bear the carbonyl group on the interstitial carbons. This thing is done. So now again, as we did that, there are categories of different categories of aldoses. Similarly, there are categories of the ketoses as well. For example, there is ketotriose, which is called dihydroxyacetone. There is ketotatarose, which is called erythrulose. There are ketopentose, which is like ribulose or xylulose. And there are ketohexoses, which are like pesicose, fructose, sorbose, and tegatose. And this categorization is again depending upon that a particular monosaccharide is made up of how many carbons, or the carbon skeleton is composed of how many carbons in that particular molecule. So we need to learn the name. We should be knowing that erythrulose is a ketotetrose, right? If ribulose is ketopentose. Now, once you know that which ketose falls in which category of ketoses, then it becomes really easy. For example, somebody says we need to draw the structure of xylulose. We know that it is basically ketopentose. If it is ketopentose, I need to draw a chain of 5 carbons, right? So a chain of 5 carbons, done. 1, 2, 3, 4, 5, right? Now, it is ketopentose, right? Terminal carbons cannot bear the carbonyl group. There is certainly a carbonyl group which is present on the second carbon, right? Now, this is the basic structure. Now, we know that rest all the carbons will be fulfilling their valencies with the help of at least one hydroxyl group. Same will be done by both the terminal carbons over here. So this terminal carbon can have a hydroxyl group in this direction or in this direction or in this direction, any direction. I am keeping on right. I can keep it on the left. On the other one, we can keep it on the left. So about the terminal carbons, to which direction hydroxyl group is oriented, it really doesn't matter. But we need to be just like in polyhydroxyaldehydes, or we can say aldoses. We need to be really careful about the orientation of the hydroxyl group around the interstitial carbons, right? So for that, we have our codes. Code for xylulose is 4. Now, 4 means just on the carbon number 4, if we number it like this, just on the carbon number 4, its hydroxyl is present on the right-hand side of the carbon skeleton, which actually also means that on carbon number 3, it will be oriented on the left-hand side. Now, fulfilling rest of the valencies with the help of hydrogen atoms in just alternatively, right? So now this is xylulose, right? If it would have been ribulose, code would be 34 on all both the positions. Like then on the third carbon also, there will be the hydroxyl group on the right-hand side only, like on both of them. So this is done. Now, there are a few questions, right? First of all, why we are drawing all these structures vertically, right? Second question is, why the orientation around the only around the interstitial carbons is mattering, why we are not so considerate about the orientation of hydroxyl group on the terminal carbons, be it polyhydroxy aldehydes, be it polyhydroxy ketones, means be it aldoses or ketoses in any kind of monosaccharides. We are just very careful. We have formed the codes to learn the orientation of the hydroxyl groups around the interstitial carbons, not the terminal carbons. Why so? The reason is here, all these structures are not just the chemical structures, they are actually the Fisher projection formulas. Now, why we are drawing these Fisher projection formulas? We should just keep this thing in mind that these molecules are not two-dimensional. they all are three-dimensional, right? They are not lying on our paper or screen only. They exist within the three-dimensional cells. So all these molecules are three-dimensional. They are not like straight chains and on one side on the straight chain, the on the straight line, others are no, it's not like that. This is just the representation. When we need to represent a 3D structure on a 2D surface, we need some kind of formulas, or we need to just find a way of representation. So Fisher projection formula, which we have been learning, it is just a representation. So it is just a representation of how a molecule can be, 3D molecule can be shown on a 2D surface. First of all, now this Fisher projection formula has some conventions. First of all, we will draw the structure on a vertical line. Now, the group with the highest priority should be drawn on the first carbon. That's why although both the terminal carbons have equal tendency to form the carbonyl group, if we are talking about the aldehydes, or any of the interstitial carbons have the tendency to form the carbonyl group. But by convention, when we need to show these structures, we don't do this, right? We just draw the carbonyl group either on the carbon number 1 in case of polyhydroxyaldehydes, or we draw the carbonyl group on the carbon number 2 in ketosis. So this is one thing. Secondly, why only the interstitial carbons? Now, the answer lies in another concept of chemistry, which is isomerism, basically. So basic thing is isomerism. Now, what is this isomerism? Isomerism is basically a comparative phenomenon which we study in chemistry. What is that? It is comparative, right? Which means that if you compare two molecules, if they have same molecular formula, then first criteria is done to be the isomers of each other, right? So they need to have same molecular formula. If they have same molecular formula, but different structural arrangement, and in some cases, even this structural arrangement of the groups is also same. So there are cases where the molecular formula is also same and the structural arrangement is also same. In that case, or spatial orientation is different. So, which actually means same molecular formula. Now, what is molecular formula? If I want to find the molecular formula of, for example, this fructose over here. So it is basically to know that what is the composition of different atoms in a particular compound or in a particular molecule. So over here, there are three kinds of elements present. One is carbon. So let's count how many carbons are there. 1, 2, 3, 4, 5, 6. So there are 6 carbons. So it is C6, right? Now, how many hydrogens are present? So 3 hydrogens over here. 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 hydrogens which are present over here. And how many oxygens are present? 1 oxygen in all of them, right? So this is the molecular formula of fructose, right? Now, let's see what is the molecular formula of tagatose. If you will count over here, here also there are 6 carbons. So it is for tagatose. What is the molecular formula of tagatose? C6. Count the hydrogens, you will find that they are again 12, right? Then now count the oxygens, you will find they are. So these fructose and tagatose are two different molecules. They have very different properties, physical or chemical, but still they have the same molecular formula, right? So it means their first criteria of being isomers of each others is done. Now, if they may have the different structural arrangement, do they have the different structural arrangement? Let's check. On the first carbon, there are two hydrogens attached and one hydroxyl group attached. Right over here on the first carbon, same thing, two hydrogens attached and one hydroxyl group attached. On the second carbon, there is double bond O. Here also on the second carbon, there is double bond O. On the third carbon, there is one hydroxyl group and one hydroxyl group and one hydrogen group. On carbon number 4, there is one hydrogen group and one hydroxyl group. If you will just see, the orientation is different, but if you will see, it can, in short, it can be, this can be written as CHOH, and this can also be written as CHOH. And same thing we can, if you just form the condensed thing, this is CHOH, this is also CHOH, right? So structurally, over here also same composition, CHOH. This is also CH, one H and one OH, right? So last terminal carbon is again CH2OH. It is also CH2OH. So it means even in the structural arrangement, there is no difference. It means if there would have been a structural difference, or there would have been a change in the structure, then it would have been a structural isomer. So now, right now, it is not a structural isomer, right now. But we know still fructose is like totally different from the tagatose. Now, what is the difference? Difference is just the spatial orientation of the hydroxyl group, right? Difference is just the orientation in the space, right? So that is the only difference in this. So if there is difference in the spatial orientation, then such isomers, so the isomers are of two types. Basically, if there is the difference in the different in the structural arrangement, then those isomers is of two types. Those isomers are called structural isomers, but we have just checked that there is not any structural variation, right? Now, if there is just the difference of spatial orientation, then these isomers are called stereoisomers, or that is called stereoisomerism. That is called stereoisomerism, which means the isomers which have same molecular formula, same structural arrangement, but their orientation in the 3D space is varying. That is leading to the stereoisomerism, that is leading to the different physical, chemical, as well as optical properties, right? Optical properties means that how much they can rotate the light, whether they can rotate the light, and if they can rotate the plane polarized light, then in which direction they are. So there are so many things. This is actually a very detailed topic of chemistry, but now point over here is that we are not gonna deal with structural isomerism over here. We just need to know about stereoisomerism. Now, what is the most important thing for stereoisomerism to occur? It is basically asymmetry. So let's do that. I told you that what is the most important thing? It is asymmetry. Asymmetry. Right, now asymmetry is what? Asymmetry is like, if what is there in asymmetry, like if you are looking at a particular thing from different angles, from different positions, then it may appear structurally totally different from different angles, right? So for example, if you are looking at a sphere or any ball, right, from any direction, it will look actually similar. Its structure will look similar. But if you will look at, for example, hand, from any one position. Like for example, right now I can just see a totally different aspect of my hand, and you can see just totally different, which means that the position of the observer and the orientation of the subject. So my hand is basically the asymmetrical subject, which it matters, right? So from wherever you are looking, if you will change your position as the observer, you will be looking at a totally new perspective, from a totally new angle, and your perspective about the structure of that thing will change, right? So when the perspective about the structure of something changes with the position of the observer, it means that that thing is asymmetrical, right? Now, what is very important for a molecule to make it asymmetrical? It is the chirality of the carbons. Chirality. There must be the presence of chiral carbons. Now, what are chiral carbons? It can be any chiral group. It not only about carbons, it can be chiral sulfur or anything, but generally it is like in biochemistry, and where this stereoisomerism and isomerism is actually playing a role. It is basically, it happens generally in hydrocarbons. So we generally talk about the chiral carbons. Now, what are chiral carbons and how they create asymmetry? Chiral carbons are those carbons which satisfy their valency, satisfy their valency, four valencies, right? Or I should say tetravalency, tetravalency, with help of, with help of four different molecules. Different molecules or groups. Groups is the better word. Generally, they are atoms also. So if it's tetravalency, means more four valencies. If it is just with the help of four different types of groups, basically types of groups, then it is a chiral carbon, right? And if it is a chiral carbon, it renders the molecule asymmetrical. And whenever it renders the molecule asymmetrical, a phenomenon of stereoisomerism occurs, right? Which leads to the creation of so many stereoisomers, right? First of all, let's make the concept of chirality clear. So, let's see dihydroxyacetone. Like this is the only, now this is a fact about this. This is the only monosaccharide which does not have any chiral carbon. Rest all the monosaccharides have the chiral carbon. So why it is not having a chiral carbon? So over here, if you look at this carbon, two of its valencies are fulfilled by two hydrogens, and then one is fulfilled by this, right? One, two, three, and then fourth one is fulfilled by hydroxyl group, right? So this is different, but this is the same. The hydrogen atom is the same kind of group, right? Because it is associated with the same kind of groups at two positions, that's why this carbon number one over here is not chiral. Now, let's go to this central carbon over here. So one of its valencies is fulfilled by this group. Another one is fulfilled by this group, which are actually the same, right? And the third and fourth valencies again satisfied with same kind of groups. So it is like so many same type of groups it's associated with. So that's why it is not a chiral carbon. Same is the case with this. If you will say there are the two H2 groups, so again, not chiral. Now, let's see some chiral carbon. So same criteria will be followed for rest of them. So let's see some chiral carbons here, not chiral, right? Because it is associated with two hydrogens, which is same kind of molecule. Again, Not chiral because on one side it is associated with this, on other side says this is also different, but third and fourth valency is fulfilled by a single molecule of oxygen, which means the same kind of group. It means it is again not chiral. So second carbon on which carbonyl group is there, that carbon is also can never be chiral. So what are the chiral carbons? Chiral carbons are like this. On this side, this carbon is associated with the whole of this molecule, right? On the other side, this fulfilling its valency with the help of this group. Then on the third, valency is fulfilled by this group, and fourth with this. So four different kinds of groups are satisfying the four valencies of this carbon number 3. That's why carbon number 3 is chiral. Same as the case with the carbon number 4. If you will look at the carbon number 4, then if you will look at the carbon number 4 over here also, one is this one group, another group is this one, third and fourth. Four groups satisfying, four different groups satisfying four valencies of carbon, again the chiral carbon. But again, this terminal carbon is not the chiral carbon because two of its valencies are fulfilled by the same kind of groups, which are the hydrogen atoms. So it is not the chiral. So because this is the criteria to find the chirality, always and always, just the interstitial carbons can be chiral. Just the interstitial carbons can be chiral. Be it the other part, like on the aldoses, we can go to that slide as well. So if you will see in the case of aldoses, just the interstitial carbons are chiral, right? Now, in ketosis, in aldoses, all the interstitial carbons are chiral, while in ketosis, because one of the interstitial carbons is involved in the carbonyl group, which renders it achiral. So generally, if there are the same number, like aldohexoses or ketohexoses, then aldohexoses are gonna have more chiral carbons than ketohexoses. Like in the aldohexoses, there will be four interstitial carbons, right? And all four will be chiral. Again, in ketohexoses also, there will be four interstitial carbons, but only three will be chiral. So that is the important point. Now, because now you must have got the idea that why orientation around interstitial carbons was actually taken into consideration to such an extent, because they are chiral. Terminal carbons are not chiral. That's why orientation around them doesn't matter. But the orientation around the chiral carbons makes difference. So we need to be very specific. So this is about the stereoisomerism. Now, all the aldohexoses or all the ketohexoses, all the ketohexoses, the four which you can see over here, they all are basically stereoisomers of each other. They all are stereoisomers of each other, all these four, right? All the ketopentoses, like there are two, they are stereoisomers of each other, right? For erythrose, that option is not available right now, but we will study. For over all these aldopentoses, they are actually stereoisomers of each other. These all these 8 aldohexoses which are written over here, they all are again stereoisomers. These two are also stereoisomers of each other, because they're oriented over here, the orientation of the hydroxyl group around the chiral carbons is varying. So this variation creates the stereoisomers, right? But as you know, they all are related, which means they all are related to one another by this phenomenon of stereoisomers. But it's like they all are siblings, right? But we know there are a few siblings who have a special bond or special relationship among themselves. Like they are all our siblings, but they are special to each other. So there is a certain, this kind of relationship is seen when we actually discuss another kind of thing, which is apimers. So apimers are what? Apimers are stereoisomers which vary in orientation around just one chiral carbon, or we can say configuration around just one chiral carbon. They are they are stereoisomers of each other, but of special kind, which we call as apimers, right? So, for example, let's take the case of galactose. The code for galactose is 25, and for glucose it is 245, right? So, which means that on carbon number 2, in both of them, the hydroxyl group will be on the right hand side. On carbon number 4, in case of glucose, the hydroxyl will be on the right hand side, while in the case of galactose, it will be on the left hand side. While on the carbon number 3, in both of them, hydroxyl will be on the left hand side because it has not been mentioned in the code of any of them, right? While on the carbon number 5, the hydroxyl group is again on the right hand side on both of them. So they are, where is the difference in the structure of both of them? It is just around the carbon number 4. Here the orientation is varying, right? So only at this position, only one position, they are differing. So they are actually, glucose and galactose are stereoisomers of each other, but of special kind, that is, they are apimers of each other as well. Same is the case when we compare this mannose with the glucose. Mannose is like 45, code is 45. So apart from carbon number 2 over here, the configuration around rest all the chiral carbons are interstitial carbons also. So all the chiral carbons is like it's exactly the same. So you can just compare, it's exactly the same, right? So um, these are also apimers of each other. But when we compare the orientation around galactose and mannose, you will notice that, okay, let's compare. At carbon number 2, they are varying, right? On this over here, it is hydroxyl group is on the right hand side, but here it is on the left hand side, right? On carbon number 3, they are same. On carbon number 4, now here it is, there is the variation. Over here it is on the left hand side, while over here it is on the right hand side, right? And the carbon number 5, it's all same, right? Terminal carbon or orientation around the terminal carbons doesn't matter. So over here, the structures of these two are varying in terms of configuration around two carbons, two chiral carbons. So that's why, due to that reasons, galactose or mannose are stereoisomers of each other. They are, no doubt, but they are not the apimers of each other. So that is the difference. So I hope now the concept of apimerism is clear to you. Now, stereoisomerism is actually the phenomenon which is based upon the chiral carbons. So due to that only if we need to find that how many stereoisomers are possible, it depends upon how many chiral carbons are present in that molecule, right? So how to do that? So there is a formula for that. That formula is 2n, 2n. N over here is number of chiral carbons. For example, this glyceraldehyde. In dihydroxyacetone, there is no chiral carbon, so it is 2 raised to the power 0. right? So 2 raised to the power 0 means just one stereoisomer, which is that one structure of dihydroxyacetone, which we have seen. Now, over here, just one, in glyceraldehyde, there is just one chiral carbon, due to which the formula will become 2 raised to the power 1. So for glyceraldehyde, two stereoisomers are possible. For erythrose, there are two. So over here, 2 raised to the power 2 means 4 are possible. Similarly, ribose, 2 raised to the power 3, which means 8 are possible, right? So in glucose, it is 2 raised to the power 4, which means 16 stereoisomers are possible, right? Okay. So it means, and fructose, again, there are 3. In fructose, there it is ketohexose, but chiral carbons are 3. So again, the case is that of basically aldo pentose, right? So it will be again 8. So this is the thing. Now let's look at that. For glyceraldehyde, two isomers, two stereoisomers are possible. Now I'm going back. Okay? We are going back to the slides to just see which are those stereoisomers. Just to see which are those stereoisomers. Okay. Over here, two are possible, but only one is drawn. Why? Okay. For erythrose, which has two chiral carbons, 4 are possible, but just 2 are drawn over here. Why? For this aldopentose, which has 3 chiral carbons, right? 3 chiral carbons, 8 are possible, but only 4 are drawn. Where are the other 4? Over here, for aldohexose, which has 4 chiral carbons, 16 are possible, but just 8 are drawn over here. What are the other 8 stereoisomers? So the answer lies in this prefix, which we have ignored till this time. Till this time, I was just reading this as erythrose, while the name mentioned over here is D-erythrose. It is not just erythrose, it is D-erythrose, D-threose, D-ribose, D-arabinose. The answer lies over here. Now, what is that? It is like, okay, there are basically, if there is D-erythrose, there is another version of erythrose, which is also called L-erythrose. If there is D-glyceraldehyde, there is another version of glyceraldehyde, which is L-glyceraldehyde. Similarly, if there is D-ribose, there is another version of ribose, which is L-ribose. So let's do that. Let me draw glyceraldehyde first, right? So what is D-glyceraldehyde? D-glyceraldehyde, what is the code for D-glyceraldehyde? So same, glyceraldehyde, I must be knowing that it is aldotriose, which means it is composed of 3 carbons, right? Now it is aldotriose, it means on the terminal carbon, 1 oxygen, right? And over here, again, there will be 2H, OH, again, no orientation problem, so we can just write it as it is. Now, on the second carbon, its code is 2, which means on the second carbon, OH group is there, right? Right. And on this side, it means on the left-hand side, H will be there. Now, this two code is 2 that on the second carbon, it needs to be on the right-hand side. But this is true only for D-glyceraldehyde. This is true for just D-glyceraldehyde. But if this is the case for L-glyceraldehyde, if I need to, which is another stereoisomer of D-glyceraldehyde, if I need to draw that, it will also have 3 carbons, because this is again glyceraldehyde only, nothing is changing, right? So again, 3 carbons, right? Again, the carbonyl group on the uppermost carbon, no problem. Again, this doesn't matter. Point is over here. This L-glyceraldehyde is also having the code 2, but this time that 2 will mean that hydroxyl group on carbon number 2 lies on the left-hand side. For L-glyceraldehyde, the code 2 means that hydroxyl group lies on the left-hand side. Now, if you will see the diagrams of both D-glyceraldehyde and L-glyceraldehyde, you will notice that they are just the mirror images of each other, right? Right has become left and left has become right, right? So they are the mirror images. So one is this kind of stereoisomers where their positions are altered just on few of the carbons, right? They are just altered at one or the 2 carbons, and all these stereoisomers are created. Then there are the stereoisomers where out of those stereoisomers, there is a special case of epimers where the position is altered just around only 1 carbon, right? Those are called epimers. That is a totally different thing. Now, over here, when positions around all the carbons are altered in one go, then all these stereoisomers, which are formed in this way, they are called enantiomers. When they are called enantiomers, when everything becomes whatever was right, it just becomes true for the left hand side. Codes becomes true for the left hand side. Then those stereoisomers, which are formed in that manner, they are the mirror images, and they are actually the stereoisomers, which are also called enantiomers. Let's do another case. Let's do another case. For example, erythrose. Erythrose, right? Or we can take anything else. Let's take glucose. Glucose, the most important monosaccharide, right? Glucose is basically aldohexose, right? 1, 2, 3, 4, 5, and 6. This terminal carbon will have carbonyl group, and this will be, there will be 2H and OH fulfilling that. And then the whole story is about these interstitial carbons. Now, what we need to do over here, we need to write the code. It is 245. Now, if I'm drawing D-glucose, then this code 245 means that on the carbon number 2, carbon number 4, and 5, hydroxyl group needs to be on the right-hand side of the carbon skeleton, which means on the carbon number 3, hydroxyl group will be on the left-hand side, and over rest of the positions are just filled alternatively with the hydrogens. Now, if I need to draw L-glucose, then code will remain 245 only, and the chart, of course, the carbon chain will also remain that of 6 carbons only. 1, 2, 3, 4, 5, 6. The terminal carbon will have a carbonyl group, this now, and this will be CH2OH. Done. Now we are coming to on 245. This time for L-glucose, it is like it should be on the left-hand side. 245, right, on OH over here on the left, which means in L-glucose on carbon number 3, OH will be on the right-hand side, right? And if this over here is on the left-hand side, then H will be on the right-hand side, H on left, H on right, H on right. So these two structures, if you will look at them, they are mirror images of each other. They are non-superimposable, right? These two are also the stereoisomers of each other. They all have the same name, right? The only structural difference over here, the properties are like very, very similar, right? Especially the chemical properties. So That's why the names have been kept same, right? The only difference which comes along is that they show different optical properties. So uh, these mirror image, these such stereoisomers are called enantiomers. Now you would say that how to recognize whether some enantiomer is D form or the L form. And remember, this we are talking about capital D and capital uppercase D and uppercase L, not the lowercase d and lowercase l, which we can say as small d or small l. It is not about that. It is totally different from that small d, small l, which goes for this small d and small l stands for dextrorotatory or levorotatory behavior, which means that this particular molecule just turns the plane polarized light either to right or left, right? It is a totally different thing. This is just the denotion. It has nothing to do with the optical properties. They are optically active stereoisomers, no doubt, but this D, capital D and capital L just is to denote that something is different between these two forms, just that. Certainly you cannot remember the codes for all of them. You should, right, at least just learn the codes for all the important ones, and all the important ones over here are marked by these rectangular blocks. These rectangular blocks are actually denoting all the important ones. But if you can learn from all of them, then it's really good. But if somehow really don't remember the code, then you need not to actually look at other positions. You will actually look at the position which is the chiral carbon farthest from the carbonyl group. For example, in this case, this is the carbon, all these are the chiral carbons, right? This is carbon number 1, 2, 3, 4, 5, right? So this is the chiral carbon which is farthest from this carbonyl group. The chiral carbon number 5, right, is farthest. So you just look at the position of OH on this carbon. If it is on the right hand side, then that enantiomer is D form. And if it is on the left hand side, then that enantiomer is the L form. So this is all about different forms. This is how. So now, if the question which arose from here was that where are the other 8 for here? So other 8 for R, these are the 8. Then other 8 are, this is the D form, L-allose, L-altrose, L-glucose, L-mannose, L-glucose, L-idose, L-galactose, L-tallose. So each form, these are just the D forms written over here. They all have their mirror image structures, which are denoted by the L versions of these stereoisomers. So total 16 are just there. Regarding the stereoisomerism, so this is diglyceraldehyde and L-glyceride, which we have actually discussed, right? So Fisher projection formula is telling us this. So they say that if this farthest carbon follows this structure, then it will be D. For example, over here, the furthest question, the orientation around this is same as this. So it will be D. And if the farthest chiral carbon from the carbonyl group shows the orientation just like L-glyceraldehyde, then it will be L. But the thing is, we need not to compare it with glyceraldehyde, DL glyceraldehyde or L glyceraldehyde. Remember, if OH is on the right hand side on the chiral carbon which is farthest from the carbonyl group, then it is D. If the hydroxyl group is on the left hand side on the chiral carbon which is farthest from the carbonyl group, then it is L. And these D and L forms occur for all the stereoisomers, for both aldoses and ketoses. With this, the basic linear structure of all the monosaccharides is done. But after this, these structures get twisted. Now, when they get twisted, they create their cyclic structures. So, in this next session, we will discuss those cyclic structures. Till then, goodbye, take care. Please revise the structures, please remember the codes, and just work on them. It's not hard. Once you start doing that, it will become really, really enjoyable. See you next time. I will wait for you. Bye bye.